Hello and welcome back to the session. In this session, we are looking into Pseudomonas. So, Pseudomonas aeruginosa is a gram-negative, non-fermenting aerobic rod. It is ubiquitous in the environment, the soil and water, and is quintessential opportunistic pathogen. It rarely infects healthy tissue but ruthlessly colonizes compromised hosts, particularly those with cystic fibrosis, severe burns, or neutropenia. So, gram Steinler, we could see it as pink or red rods, as we have portrayed here. It's highly motile via a single polar flagellum. It produces pyocyanin, so it's basically a blue-green pigment. It generates RO species, and pyoberidin, that's an yellow-green pigment, a siderophore. So order Pathona, it has a distinguished fruity or grape-like corn tortilla order. So Pseudomonas is like an adaptive guerrilla force. It does not need a specialized base. It can live in dirt or water, waiting for a weak point in the border after the host skin or lungs to strike. So Pseudomonas possesses a massive arsenal of weapons. Exotoxin C. So it basically inactivates elongation factor 2 via ADP ribosylation, stopping host protein synthesis. A proalginate, an exopolysaccharide that creates a thick biofilm, and this protects the bacteria from phagocytosis and antibiotic penetration. Phospholipase C degrades host cell membranes, specifically surfactants in the lungs. Pyocyanin directly kills the competing bacteria and host cells by creating oxidative stress. So it uses pili to attach to the damaged epithelium. The bacteria talk to each other via chemical channels. Once their population reaches a certain density, they simultaneously activate toxin production and biofilm synthesis. And in cystic fibrosis patients, the bacteria switch to a mucoid phenotype, creating a permanent fortress in the lungs. While the host sends neutrophils, the biofilm and exotoxin neutralizes them, leading to chronic inflammation and tissue destruction. So here, once the guerrilla force enters, they don't just stay in open. They use allogeneic concrete to build a fortress. That's our biofilm. They use radio signals, the quorum sensing, to coordinate a massive simultaneous ambush, so the host's border patrol, or neutrophils, cannot reach in time. Laboratory diagnosis, culture. So it grows on blood agar and McConkey agar, as it's a non-lactose fermenter. Oxidase test positive, and this particular test distinguishes it from the Enterobacteriaceae. It requires a specialized antibiogram because Pseudomonas is naturally resistant to many common drugs. Prevention and prophylaxis. In hospitals, avoid keeping flowers or stagnant water in rooms of neutropenic patients, as Pseudomonas thrives there. Rigorous sterilization of ventilators and catheters, which are common sites of biofilm formation. Treatment, Pseudomonas is notorious for multi-drug resistance. So treatment usually requires an anti-pseudomonal agent, often in combination. So piperacillin or tazobactam, it's basically an extended spectrum penicillin. Ceftazidime and cefepime, third or fourth gen cephalosporins. Carbapenems, meropenem. Fluoroquinans ciprofloxacin, aminoglycosides amikacin or tobramycin. So B pseudo அப்படின்ற ஒரு நிமோனிக் ஞாபகம் வச்சுக்கலாம். Burns, endocarditis, IV drug usersல, pneumonia, cystic fibrosis இருக்கிற peopleல, sepsis, external otitis, அதாவது swimmers ஓட earsல, UTI catheters யூஸ் பண்ணுறவங்கள, and people who are drug addicts, and people with osteomyelitis, அதாவது puncture wounds through sneakers and stuff like that, இது வந்து ரொம்ப common. So Yeah, that's it for pseudomodal infection. Please do read Apurva Shastri once to correlate it with the text.